Hello. All right. Good morning and welcome everyone to our Everglades seminar series highlighting our faculty expertise and their programmatic uh, research. Today we have with us Dr. Katia Xavier and the pathology team who's going to talk to us about vegetable and sugarcane pathology, current findings, and future directions. With that, Katia, the floor is yours.
Thank you, Django, for the introduction. And good morning, everybody. So, yes, I'm happy to be here and to be talking a little bit about what our lab is doing. So, to start with, not passing the slides. Let's see, I'll try the control. I think I need some help, but I can keep talking. so I'll try to keep talking. Anyway, I started this position almost 3 years ago, and as the plant pathologist here, but I don't want to actually talk about plant pathology. I want to talk about healthy plants, because when we go to the supermarket, what we want to buy is like beautiful, vegetables, beautiful sweet corn, and stuff like that. I wish I had my picture to show in here, but just imagine that you're going to do your groceries now and what you want to find at the supermarket, okay? But for the growers, that's not quite the reality. For them to be able to produce nice, beautiful vegetables and to sell it to the stores, it's not that easy, it's very complicated. And one of the causes are, are diseases because the pathogens are out there. So just like us, the plants can also get sick, right? So when we are sick, what do? We go to the doctor, right? So when the plants are sick, they also need to see a doctor. But it's not quite the same because, and I still don't see my slides. I need my guys to help because it's working. Okay. Because I have not memorized the whole presentation. But anyway, so the plants also can get sick and they need to see a doctor. And there are several ways that the plants can get sick. Like we can get a cold, right? We can get respiratory diseases. There are several ways that we can get infected, and the plants is the same thing. So and in here I'm just showing 2 cases because that's the most important for this presentation. But let's say if the plants get, there are some airborne pathogens, so the spores can land on the leaf surface and they can infect the leaves and cause spots like on the first picture here on the top left. And in the end, what the growers will see if the severity or the levels of this disease is too high in the field are corn ears that look just like this. So it's not beautiful. It's not going to produce high yields to the growers, and it's going to cost the growers a lot of money. So they don't want to see that. In this case, on this lettuce field, what happened is that this is a soil-borne pathogen. What that means? It means that this soil can survive this pathogen can survive in the soil, and then it's going to infect the roots. The roots, they're in the soil, they need to get water and nutrients and then transport that to the leaves so the plant can grow beautifully. But if the pathogen is there, it's going to block that. So it's going to be a big loss for the grower. And on top of that, this pathogen specifically can survive in there for years and years and years. So that's a big, big problem for the growers. So because of all these diseases, we have this beautiful, great team of plant doctors here in the EAA. So what this team can do to actually help the growers to manage this plant disease, how they can help them to let's say, control. We don't use the term control because most of the times you can actually cannot control. We manage it. We can bring the level of that disease lower, and then the grower can still produce and make money out of it. Sometimes we cannot completely control that disease. So let's go to the next slide. But I put some pictures here for some reason, sorry. So one important step that we have to do is to go to the field and observe the disease in there. And then what the growers can also do is to bring those diseased or sick plants to our lab, and we can help them. Because that's actually the most important step, is to do the correct identification. So when we go to the doctor is going to run a lot of tests on us because they are trying to identify what's causing or what's making us sick. If you guys have kids, you know exactly what I mean. You bring them to the doctor. If it's a respiratory disease, they will do strep. Kids will cry. They will also collect something in their nose. They will also cry. And then they'll run some tests. They want to know if it's COVID, if it's a strep. They will take blood to run some tests because it can be some virus as well. Why? Because they have to give the right medication. If it's a bacteria, right? They want to give antibiotics. So in our case, we also want to identify it correctly so we can tell the growers what they can do against that disease or that pathogen. So that's why I made this slide, because from here, I was talking about identification here. We have identification, and then we want to do the correct identification to come up with the management strategies for the growers, right? And we have a lot of steps to get in there. We do a lot of research to help the growers to get there. But in our case, for this presentation, and actually what my lab is doing, is a lot of, we have a lot of projects here at this step, and some that are here at that step. So why? Why is that? Because we have a lot of emerging diseases, so we have to do the identification. Why? Because the disease is new in here, so we have to do the correct identification. Or there are some diseases that were actually identified here, but they were identified back in the day. So that means what they used to identify the disease was morphological characters, or yes, morphological characters or based on the symptoms. And then they did not use molecular tools because maybe the molecular tools, they did not even exist by that time, okay? But we still rely on that identification. So what my group is doing is to now go and use molecular tools to do the correct identification of some of those diseases. And for diseases that were already identified using molecular tools, we are one step ahead, I would say. And then we can go and have a plan or a project and start to study some management strategies that are applied here for the EAA. Sometimes there is a lot of research and they're using it there in the Midwest, let's say, right? Like for corn disease, there's a lot of research in there. But the problem is sometimes the strategies that they have in there do not apply in here because our conditions are very specific. One thing that makes it very specific is the type of soil that we have. I need of water, I don't know. Yeah, the type of soil that we have. So that's why we have to study it in here, because we want to make sure that our growers will have the information that's adapted here to our area. Okay, so we have a lot of projects going on in the lab, and Tarciso, Larissa, and Fernanda are working on either emerging disease or some diseases that were identified back in the day. And then Larissa, Alba, and Vitor are working on diseases more like on management strategies for some of the diseases. Now, sometimes those diseases were identified, and we rely on those identifications that were even identified based on molecular tools. But what happened is that we are finding some, how can I say, it's like, let me pass this slide and you understand it, right? So it was identified based on molecular tools, but we are finding out some new stuff. And you think, okay, great, we can go and do the management strategies and help the growers. But you have to do the identification again, because now we found out that that disease that was very well known in here in the Midwest, we found out there are several other pathogens that are related to that same disease. So now we have to redo the identification of that same disease. So this is what happened with Victor's study. So he's going to talk more about it. So I just want to give you some overview so you can understand the projects that they will be talking about here. Overall, this is how we do morphological identification. I wish I can show it here. So we collect samples in the field, bring them to the lab, cut little pieces, surface sterilize. I'm not going to go into details. Plate it on media, look under the microscope, and check for spores and see how they look like, and go to the literature and just check and say which type of fungus that is. And then we take that one single spore, grow it. Oh, sorry. And then grow it, extract DNA, do PCR, that's the molecular characterization. Run PCR with specific primers, send it for sequencing, and build phylogenetic analysis, okay? I'm not going to go into detail, I just want to give an overview of that, because most of my students that will be talking about, will be talking here, they did that, but they are not going to go into details. So those are the projects we are doing in my lab about sweet corn, celery, lettuce. We have some on sugarcane too. So Izuri, she's supporting our team. She has been a great helper. She started in the lab in August of this year, and she's going to talk a little bit.
Good morning. I My name is Isuri Palma. I'm from Nicaragua. I started working at the plant pathology lab in August 16th of this year at the invitation of Dr. Katia Xavier. June, I would like to thank as well as the entire lab team. My job is to support Dr. Katia and her students with their research experiments in fire, greenhouse, and lab activities such as planting, irrigating, and monitoring plants. Applying the necessary treatment, taking plate measurements, and other things. I'm learning a lot with this job and have the opportunity to meet many good people for my personal and professional growth. Thank you all.
Thank you, Izuri. So we are very proud of Izuri. She only speaks Spanish, and she did a great job now speaking English too, so she's learning. So with that, now I will invite, let's see. Yeah, so the first project we are going to talk about is Northern Cornleaf Blight. This project was approved by FDEX. It's a collaboration with Dr. Marcio Rezende and Anna. And is Vitor connected? He's ready?
Yes,
So,
I am here.
going to talk about it. Vitor is online. He's in Brazil, so he's going to present in this part. Is he in control now? Vitor,
I requested control. You have to accept.
Avery or Lucas, can you please give him control?
Go ahead, Vitor.
Okay. Oh, whoa, whoa, whoa. Sorry. Oh,
Okay.
we go back.
Vitor, just let me do it.
Okay.
Okay, now you have it.
Okay, so hello everybody. I am Vitor. I'm sorry that I'm not able to do it in person, but I'm glad to share some of the work that I've been doing this past few months. To start with an introduction, I am currently working with sweet corn, an important vegetable grown in Florida, where the major producing areas are located in Palm Beach and Miami-Dade County. And when we talk about South Florida, where these counties are in terms of environmental conditions, what we have almost year-round is high humidity and warm temperatures. Just perfect conditions for diseases to develop. And out of these, Northern Curly Blight, or NCLB, is highlighted as the most important disease in South Florida on sweet corn. NCLB is caused by a fungal pathogen named Extrahilarm tursicum and appear as these large cigar-shaped lesions that can turn into gray with time, as shown by the image here. Okay, now going to the problematic. Some sweet corn growers in South Florida have reported their difficulty to manage Northern Corn Leaf Blight, or NCLB, despite deploying fungicide with different active ingredient and as well increasing the number of applications. And for that, there are two possible hypotheses. Either a new race has emerged in Florida, or the population of the pathogen has become resistant to the fungicides used in the region. And to address this issue, we first need to understand the pathogen population across Florida, which is unknown. And that's what we are doing. We are collecting Northern Corn Leaf Blight isolates all over Florida from corn, not only sweet corn, but also field and Criollo corn. Most of our isolates are from Palm Beach and Miami-Dade County, but we also have isolates from Henry, Martin, Suwannee, Columbia counties, North Florida, and all this to have a fair representation of Northern Corn Leaf Blight population from Florida. So we went over Florida, collected leaves infected with characteristic lesions of NCLB, performed isolation, as Katia said, and when doing the molecular characterization, we found 4 species associated with NCLB, not only the original E. tursicum as cited in the literature. And these species include the original E. tursicum, present in 18% of the sample, E. restradon and Bipolaris yamadai with 6% both, and Bipolaris maydis with 18% and Bipolaris maydis with 70% of the samples. And this was very unexpected. Bipolaris maydis, is the original cause and agent of southern corn leaf blight, another foliar disease of sweet corn. And it appears as these small 10 lesions, as shown by the second image, very distinct diseases. So, and this association does not mean that they are causing disease. So the next step is to inoculate these isolates in healthy plants and to prove it. And that's what we did. These are pictures of infected leaves. And I included also a positive control of Bipolaris maydis caused, isolated from southern corn leaf blight. And in summary, as I am showing here in the picture, all of them were able to cause disease with different severity rates. But what I want to point out here is how different the lesions caused by bipolaris made is isolated from northern prolific blight in the top here, the 70%, from the one that is caused southern prolific blight in the bottom, the positive control. See how different they are, the lesions are. And ultimately, our results suggest that here in Florida, northern prolific blight is caused by a complex of microorganisms, not only one as cited in the literature. Okay, so now we found these species in association with NCLB. They're all able to cause disease. Now, to follow our objective and test one of our hypotheses, we need to test if our population of NCLB has become resistant to the fungicide applied. We started first with propiconazole, a fungicide heavily used in our region, and performed an in vitro sensibility test. This technique is composed of using artificial media for the fungus to grow, amended with fungicides in different concentrations. We put the fungus in the center of the plate and see if there is a reduction of fungal growth within the different fungicide concentrations. And a sensible isolate to fungicide will perform just like as shown in the picture here in the last picture. The higher concentration of fungicide, less fungal growth will be observed. And just for initial trial, we tested one representative isolate of each species found in association with MCLB. Now, the results of it. This is a relative growth of the fungus compared to the control in the different concentration of fungicides. The concentration is in the x-axis and the relative growth in the y-axis. And based on fungal growth, we were able to characterize the isolates in two groups, the resistant and sensible to fungicides. And both bipolaris made from northern prolific blight and southern prolific blight are within the resistant one, while the others are in the sensible ones. And remember, this is just one isolate per species. We're gonna test in bulk for each species. And starting with E-Turcicum isolates, doing sensibility tests in bulk by starting with the E-Turcicum species, 13 isolates. These are the results. We were able to differentiate in three groups: the sensible, the ones that even low amount of fungicide reduce fungal growth, the moderately resistant, a middle term sensitivity, and the resistant ones, the ones in which the fungus was able to grow even the highest concentration. And these two resistant ones in the graph, the top ones, are all from Palm Beach County, collected from one from field corn and other sweet corn. So to summarize, we have 4 species identified in the northern prolific blight lesions. And this high diversity could explain the lack of fungicide control. And then our fungicide test also shows that we have isolates resistant to fungicides. And for fungicide and for future work, more isolates and chemistry will be tested on fungicide sensitivity. And our ultimate goal is to improve fungicide recommendations for Florida growers. And with that, I would like to invite Dr. Larissa Ferreira to talk about her work. Thank you, everybody.
Thank you, Vitor. So today I'm gonna start talking about our tar spot project. This project, which Vitor was also involved, was funded by FAR, and we developed in collaboration with Dr. Marcio Rezende. So tar spot is this disease right here, which looks like tar splatter on leaves of corn, and it's caused by a pathogen called Phylacra maydis. This pathogen was introduced here in the United States in 2015, so it's a very new disease, or what we call emergent pathogen. And there is not a lot of information about this pathogen, and that's why we were awarded with this grant to study this disease here in Florida specifically. We compiled everything that we learned into an EDAS publication, which you can find here. And one of the first things we did to understand this disease was to do a survey in Florida and find where the disease was present. Because when we first started, a lot of people have heard of it, but very few people has even seen the disease. So even Anne right here helped us do the survey. So thank you very much for everyone that was involved on this process. And what we found was some hotspots, specifically here in the Homestead area, where we could find tar spot more frequently. And we also found that during the summer times, it's more likely that we find this disease. And one thing that is interesting about this disease is that tar spot or Phylacramatus forms this structure where it contains the spores, which are shown in this figure here. And these spores, that's what causes the disease. But we also found other fungi that are associated with this structure, which is called tar spot complex. And this complex of other fungi may impact disease progression, but we don't know what are the factors associated with that here in Florida. So to answer that question, to find out what are the fungi associated with tar spot in Florida, we did a sequencing of the DNA from samples collected from symptomatic tissue and also from asymptomatic tissue. And we found a plethora of fungi that are associated with tar spot. More specifically, we found 142 different fungi associated with symptomatic tissue. And these, on this picture here, I'm showing the most abundant ones. I would like to highlight that they are color-coded according to their family, and Pleiospiralis, which is an order, was the most prevalent order. Secondly, I would like to highlight that we did find Phylacramatus present in both tissues, both symptomatic and asymptomatic, and we found 16 different sequencing variants of the DNA of Phylacramatus. So through an integrated analysis, we found that this diversity of DNA, thankfully, is significantly considered low diversity. This is great news for growers because imagine that the population of this pathogen in Florida is considered homogeneous. So therefore, the management strategies that we can apply can be generalized through the state. Now, I was also interested in understanding how these different fungi correlate with each other. What are their relationships with each other? And we can see in these two networks here from the symptomatic versus the asymptomatic tissue, the symptomatic is much more complex. And guess who is in the middle of my network? Look at this guy right here in the middle. We found that Exoherillum, which is the fungi that you just learned about from Victor. We found that this fungi has both positive and negative correlations with the tar spot microbiome. This is very interesting because now when you talk about disease management in corn, we should think more holistically and consider all the pathogens that may be having some interplay with each other. With that, I'm gonna go to my next project. In 2 minutes, I'm gonna talk about Cercospora, a celery. This project is a collaboration with Anna and some other co-PIs from Gulf Coast and TREC, and it was funded by SAR. So celery, we have two different production systems, organic and conventional, and in both of them, the most important foliar disease is called Cercospora or early blight, and you can see how bad this is. If you see this seller in the supermarket, you would not buy it. So the problem is that to control this disease on the organic system, there is not really an effective control for it. And on the conventional side, they use fungicide sprays on a weekly basis. Either way is not sustainable. So this is where we come in. Our solutions for this organic system would be to identify biological controls, and for the conventional side, we're also working with AI and some drone technology to identify precise timing and monitoring of this disease. So for this presentation, I'm going to focus on some results that I have for the identification of biocontrol agents. To do that, I have done isolations from leaves and from soils, here from the ERC and also from farms. And how I do the testing to see if the biocontrol is good or not, I plate Cercospora here on two sides of the plate, and then I streak the bacteria in the middle. So this is what I got. My control here on the left-hand side is growing beautifully, whereas when I have some bacteria in the middle, it doesn't grow almost at all. So this is a around 90% reduction. Thankfully, we have 12 very good candidates that are showing more than 50% reduction that I'm very excited to test next on the greenhouse and hopefully develop a biocontrol product that all the growers here in the region will be able to use, because these are native isolates. So they're already adapted to grow in the region, and we hope that with that, our chances of success with managing this disease later will be much higher. Now, I would like to invite Alba to talk about her project.
Yes. Good morning. My name is Alba Myers, and today I'm going to talk about my master project that is going to be Fusarium on lettuce. This project is being funded by Florida Department of Agriculture and Consumer Services, and with the collaborators of Dr. Katia Xavier, Germán Sandoya, and Ana Mesaros. The title of my project is Sustainable Solutions for Fusarium Wilt Impacting Lettuce Production in Florida. The first report of Fusarium osteosporum was done by Dr. Germán Sandoya and Dr. Reid in 2017, and they did some molecular work. Then my main focus of my master project is disease management. Then what is Fusarium wilt? Fusarium wilt is caused by Fusarium osteosporum forma specialis latusae race 1. It's a soil pathogen, and born soil pathogen, and then it attacks the young plants of lettuce. And one way to describe, to see or to key out this disease is by cutting open the root system. And if you see, and when you see, let me go back. It's going forward. Can you please? Yeah. And when you see, when you cut open the half of the root system, you see some yellowing, some brownish, this one, some brownish discoloration. That's how you key out that you have a Fusarium wilt. Then Fusarium wilt is caused by Fusarium osteosporum forma specialis latusae race 1. And for saying that, my objective number 1 is to develop a bioassay to determine Fusarium of all that we call concentrations in the soil. When my project, this objective number 1 is going to have 3 experiments. At the end of these experiments, we're going to go to the field and look at these infested lands, and then that it has a low production. And then we're going to tell the growers, you need to do some crop rotation with 1 or more crops per year. Then after that year, we're going to tell the grower if it's going to be okay to return with lettuce to the same land for him to be, or for them to be to have a low infested land or with high production. For the objective number 1, we're going to use exactly the same design. That is going to be a control, but with lettuce, no with celery. The control and 10 to the 6 solutions, or how do you call that one? Dilutions. 10 to the 6 dilutions to see if to determine the concentrations in the soil. Then for the objective number 2, quantifying the effectiveness of rotational and cover crops in reducing the fusarium in the soil. We're going to have the infested land. We're going to get some rotational and cover crops like cabbage, radishes, arugula, corn, some hemp, and some sundan grass. And then it is a lot of it is a lot of literature on corn that it says that it will cause more propagules of fusarium in the soil when it's infested. But we have to do it here as well because this is done in other states, and then Florida has a different soil that is muck, and then in different environmental conditions, then we have to do it anyway. Then what we want to do is we're going to grow these crops in the field, and then we're going to see if these rotational crops are going to be equal, is going to be greater in propagules, or is going to be lesser. Then like that, but just at the end, for my conclusion, is like we're going to find out, we're going to make some, how do you say that? Okay, we're going to do some recommendations, by the way. We're going to do some recommendations and see if we can get some diagrammatic scale that the growers can come, and then growers can say, okay, I have this disease, and then we're going to say, okay, you have this amount of propagules in the soil, and then this is the way that you're going to use. You're going to use this kind of crops to reduce the amount of propagules in the soil. And then like that, we can give the growers some sustainable options to grow lettuce again. Thank you. The next person that is going to present is Tarciso Ferreira Jr., visitor scholar that he was with us in July 2023 and 2024. Tarciso, you're in.
Thank you, Alba. Can you hear me? Just confirm that you can hear me.
Tarciso, can you hear? It's frozen. Just 1 second. He's connecting. Probably it's his internet. Let's give him like 30 seconds or a minute. If he doesn't connect, I think I can cover. Do you want to present? Okay, we can switch. Fernanda can present her part, and then Tarciso can present his part later. Okay.
I'm back. Can you hear me now?
Yes.
Oh, yeah.
Okay.
That's fine.
Do you want to go ahead and present?
Yes.
Okay.
Yes,
Thank
sure.
you, Tarciso.
Thank you, Katia. Thank you for the opportunity, even though I'm not officially a LabX member anymore. We still work side by side, and it's a pleasure to me to be here. Today, I will talk about my 2 projects regarding soil-borne pathogens affecting celery and also other leaf green vegetable from the same botanical family, the Apiaceae. And also, I will talk about soil-borne pathogens affecting sugarcane. Can you... Thank you. So starting from the project regarding Fusarium on celery. It was funded by the UFIFAS, and it was done in collaboration with Dr. Katia and Ana Mezaros. You can, thank you. And this research was, starting from the good part, was the first report and description of this pathogen in the EREC. I know that it's not good for the growers, but it's good to know they're present because it's one of the most important pathogens that affect celery production in the United States. So it was the first report of this race here, that is Fusarium cispiriformis specialis API race 4. And we identified this pathogen based first on the symptoms that is showing in this picture. So we can see there are many gaps in the rows. We also can see that some plants are dead. They are dead by wilting. They're showing the image B. Thank you. And also in C, we can see a typical symptom of Fusarium infection, that is the vascular discoloration of the crown. If you notice this orange color, this is not typical in a healthy plant. So the next step was to do the molecular characterization. For that, we did a phylogenetic analysis, and we confirmed use specific primers for the race. So it's showing this picture that all the isolate that we have collected from this field test positive for Fusarium cispiriformis specialis API race 4. We have also assessed the pathogenicity, but first I would like to say that it was a really good opportunity to me to do to learn how to do a real research, how to publish, and how to write a good paper, but it was done in collaboration. It will never be possible to do alone. So I would like to thank you, Larissa, Vitor, for the previous work. And actually, he started to work with this before me, getting the EREC, and also, of course, to Dr. Katia that helped me during each step of this research. Now you can move on. So just to show you the pathogenicity test that I mentioned before, we inoculate healthy plants, healthy celery seedlings in the greenhouse, and we were able to see the very same symptoms that we saw in the field. So we could see vascular discoloration in the bottom. We also could see that the plants were not, that the fungi were able to inhibit the plant development. So the plants are stunted. They're not have the same height as the healthy plants, the control in the left. And we also could see some level of fluorosis. There is also a typical symptom of Fusarium. But a new find of this research at we have tried to see if these pathogens were able to cause disease in parsley, because parsley is frequently grown in same areas that is grown celery, and they belong to the same botanical family. And this is the first report of this pathogen causing disease on parsley. And as is showing this picture, it was really aggressive. And also in the bar plot in the bottom, we can see that the vascular discoloration, a typical symptom of Fusarium, were significant when the plants were inoculated. So we try to see if this part, that the thing that we found, that this like parsley that belongs to the same botanical family, that celery, it was affected by the same pathogen. Trying to investigate this further, we went to the field again. We did more sampling, but not just on celery. We also collect parsley and cilantro showing the similar symptoms like wilting, chlorosis. And we use selective media to select just the Fusarium that was associated with these disease plants. And we could identify 4 distinct clades besides the 4 race 4 that we have already described in the publications. But we could now, in the second survey, we could identify the race 2 of this pathogen for Fusarium cispiriformis specialis API that is less aggressive, but is also important because it's able to cause economic losses. We have also identified Fusarium cispiriformis, generally not specifically related with pathogenicity, but we have also identified Fusarium commune, Fusarium solani, and the last, we identified Fusarium pernambucanum. Interesting about this one, it has first reported in Brazil in association with insects. And it's another problem regarding celery production that being affected by the borer, the celery borer is a beetle. And this pathogen was first reported in Brazil in association with the lesions that is caused by this insect. So this is interesting, and it's just another door that can be opened and explored regarding the pathosystems of Fusarium affecting Apiaceae. But I will not gonna talk about this today. I will just try to explain what was our intentions with selecting these isolates of Fusarium. The idea was to assess if it's like Fusarium cispliformis specialis apiae race 4, other type of Fusarium associated with Apiaceae, Apiaceae crops could cross-infect the same host, which means that maybe Fusariums from these distinct clades could infect the same host, even though they were selected from a different host. Like, can you move from the next slide? I think it would be better to explain here. Here you can understand better the idea of these side projects. The cross-inoculation was to assess if isolates from celery, cilantro, and parsley could affect when inoculated just on celery, they would be able to cause disease. So we did that. We select 2 isolates from celery, 2 from cilantro, 2 from parsley, and then we inoculate all of them first on celery in 2 cultivars, then on parsley for 2 varieties. And that's we're gonna do, not me, but upcoming member for the Lab X. We will continue doing this research with cilantro. We're also doing repetitions with celery and parsley. But so far, we have some results that are interesting, that we could see that all isolates, even though they are not belong to the same host, they were able to cause disease. So isolates from celery were able to cause disease on parsley, and isolates from cilantro was also able to cause disease. This is interesting and important if you start to think about disease management, because if they are grower in the same region, in the same areas, and they can cross-infect the same host, maybe the growers should keep this in mind if they are thinking to grow celery after parsley or cilantro after celery, and so on. Can you move for the next one? Just repeating what I just say, this is important to think about disease management, and also to plan the rotation schemes inside the farms. And also, we have identified 4 new species of Fusarium associated with wilting and chlorosis in Florida that were never reported before. And we have a lot to explore, even more regarding the association with insects that I mentioned before. You can move on. Okay, now switching for the next project. Sorry if I'm going too fast. I have a lot to say in just a couple minutes. But this is my second project, not less important. It was funded by the Florida Sugarcane League. And you can move just from here. The idea was to survey a representative area from commercial fields to identify the soil-borne pathogens associated with sugarcane gaps, or the packs, that is frequently seen by the growers in the fields. And also to understand how these pathogens were geographically distributed. We know that there are many soil-borne pathogens present in the EAA, but we do not have updated data about the identity of those pathogens and how they're distributed. So from 600 isolate that we have collected and identified based on morphology, we could identify ceratocystis in around 40% of them, and Fusarium in around 30%. We could also identify different type of fungi, but it was not the intention of the research, because they were not associated with soil-borne disease. So let's focus just on ceratocystis and Fusarium. Here we have as example of one of the farms that we have assessed the geographic chord. We have combined the geographic coordinates of each sample with the associated pathogen in that sample. And with that, we could do this heat map that estimate the pathogen's distribution, or at least the disease distribution within each form. In this case, we can see the Ceratocystis and Fusarium distribution are overlapping in the field. But this was not all the cases, and more intriguing is the second one that is very we saw this very often. Let's West area is another farm in a different location, and we could see that Ceratocystis and Fusarium were located in opposite sides of the farm, which are interesting if because we can raise many hypotheses from that, like such as association with soil types that can vary within each farm, and also association with variety that can also vary within each farm. That's where we are testing now. This is a ongoing research. This is just the results of the sampling and the geographic distribution that could assess. And you can move. And here is just the pathogenicity test that we test in the greenhouse, just to show you that we have not just assumed that the pathogens were pathogenics, they were virulent. We have actually tested and confirmed that our isolates from ceratocystis, at least, we are also doing on Fusarium. We are going to do on Fusarium, but we already have the results that are showing now. From all the isolates that we collect from ceratocystis, we selected 12 and we tested in the greenhouse, and they were able to cause inhibition of the sugarcane stalk germination and development. And we have tests for two variety, the most important ones, one of those at least. And okay, with that, we have test representative isolate from ceratocystis and Fusarium. We are still doing now repetition from this experiment, and we are testing also the correlation with variety and soil types. And we also going to do a sensitivity test for propiconazole, and to see how these pathogens respond to different concentrations of fungicides. And with that, I conclude my presentation. Now I will invite Fernanda to talk about red rot on sugarcane, to explain more about Coleototricum. And the floor is yours, Fernanda. Thank you.
Thank you, Tarciso. Hello, everyone. I'm Fernanda. Okay, sorry. I'm Fernanda. I'm working with Dr. Xavier since July of the last year. And the first project that I worked was with red rot on sugarcane. Red rot is a disease that affects the stalks of sugarcane, and this causes a lot of losses in the sugar industry because they affect the sugar quality and quantity. So I think it's better. So the main symptoms of this disease is the reddish on the stalks, and in the leaves, the reddish with a pale color in the center. The main causal agent of this disease is the Coleototrichum falcarum, but in Florida, the only study doing the identification of this pathogen dates back 1984, and it's not reliable, as Katia said. So it's necessary to do the molecular identification. We did that, and using a phylogenetic analysis, and we confirmed that Coleototricum falcarum is the main agent causal of red rot in this region. And also, there is not studies about the plant pathogen interaction in the foliar phase in sugarcane. So in this photo, we did that using microscopy to elucidate it. So in this photo, I show you the foliar and have the two models of the interaction, plant pathogen interaction. The first one is the A and B will lead to disease when the pathogen, this is the hypha, the primary hypha, when the pathogen is able to colonize more than 2 cells. And in here you can see the primary hypha and the secondary hypha that indicates the colonization. And the second one will lead to no disease, that is when the pathogen is restricted to 1 cell. The hypha is restricted to 1 cell. Or even it isn't able to do the infection. Here we can see this orange halo that indicates that the pathogen tries to tries to infect the cell, but the plant has some response. So doing that, using my microscopy, we were able to elucidating the foliar phase of the disease cycle. And that starts with the spores germinating and colonizing here, and colonizing the cell, and finish with the sporulation of the pathogen in the top of the leaf. Other steps were to make sure that pathogens that we used were able to cause disease. So we did the pathogenicity test in 3 varieties from the region, 1040, 1252, and 2143. And we compare with a control with water and with a non-pathogenic isolate. So the pathogens were able to cause disease with different levels of virulence. Also, we could observe that the varieties 1252 and 2143 showed a greater resistance compared to the variety 1040. So in conclusion, the Coletótrico falcarum is the mainly causal agent of red rot in sugarcane in this region. The varieties 1252 and 2143 showed greater resistance, and these findings will be available to the management this disease. Thank you, everyone.
Thank you, everyone. So I'll just conclude now the presentation. Thank you, everybody from the lab that presented. So I started this presentation by, asking the question, how can we help the growers to manage plant disease? And I said how important it is to do the correct identification, and all the projects we are doing, they presented and they show that. So by doing the correct identification, we can come up with the management strategies that are focused here in our region for the growers. Now, there is another very important part, and 2 minutes to finish that, but it's really important to do the outreach, right? We have the information now, we can bring it to the growers. My group is doing that. So we do 101 visit at the farm. We go, we talk to the growers, we talk to crop consultants. We have a very good interaction with the extension agents, so they're all helping us to identify the problems and also to go there and disseminate the findings. Here during local growers meetings, this was actually here at the 100-year celebration in our station. We also go and we bring this information to the little ones at the schools, at the fairs. So, and thank you, Django, for being organizing that, the extension agents. We also go to conferences, plant pathology conferences, and we spread the information that we are getting here. There was one that we went that they actually said, oh, wow, we are learning a lot about this EAA. So we are spreading the information. So that's really good. We are very proud of that. And also, we are publishing the information, the findings that we are getting here. We have 2 Ida's publication, one was already published, the other one was accepted. It's in the final phase, so soon you guys will be seeing it online. We have two research papers. One was accepted, the other one is already published. So we are very proud of the work we are doing it. We do it, we love it. So doing research here is like the paradise, very humid, the muck soil, a lot of pathogens, don't tell the growers that. But in the plant pathology side is really good. We feel sorry for the growers with the hurricanes, a lot of water, but at the same time, we are here to help them, you know? So yeah, that's the work we are doing here. With that, I would like to thank my lab. They give me a lot of support, so it's a great team. We communicate really well, so I'm very proud of them all. Izuri was talking in English today. It was the first time I heard her speak in English. We are we are all also learning Spanish now in the lab. So I would like to thank all the collaborators. You know, without them, of course, this wouldn't be possible. And the growers from the region, oh my God, they help us a lot of support from them. And also, you know, I'd like to say we are always here to help them. And the crop consultants, they are great. We work together with them, the extension agents, you know, and everybody. So thank you for the opportunity to be here today and to present this seminar. So if you have any questions, I think first we are going to ask Zoom, if you guys on Zoom have any questions.
Yes, I have a question. How big of a problem is red rot in sugarcane from an economical perspective?
So here in the EAA, it's not much of a problem because I believe based on the fungicides that they actually spray to control the rusts. Probably it's doing the job. And also here in the EAA, we burn sugarcane, so that helped to control it. Coletoticum likes that crop debris, and since we burn, it's not much of a problem. Now in regions like in Brazil, they stopped burning, I think 20 years ago, and coletotic was not an important disease when they were burning sugarcane. But now that they don't burn, there is a paper reporting that coletotic is, I think, the top one disease in Brazil. So it has switched after they stop burning.
So it could be an up and coming disease once we stop burning sugarcane.
Yes. Any other questions online? Questions here in the room? Django has a question.
The slides had the heat map, and
Yes.
the unit was kernel density. Is that the kernel mean the disease kernel? What is kernel density?
No, that
You know,
the kernel was not for disease. Tarcísio can tell you more because he actually worked on that, but I'm not an expert on that. But so that's based on the points where we collected it, and not if it has disease or not. So it was collection points, and so we did not correlate. No, we did correlate it with the disease, but the important thing there was to do the points, collection points, if that makes sense. Okay. Tarcísio, are you online? Do you want to add anything?
Yeah, kernel. Can you hear me?
Yes.
Kernel density is like kernel is just the name of who described the analysis for the first time. KDA is the name of the analysis, and then he combined the binary data with the coordinates, and then he how do you say this? Like he's help me catch with the word I forgot. Like as
Yeah,
the
say in Portuguese,
project
I'll try to translate.
is like to ele
Like
prevê prever a posição do patógeno?
Try to estimate. Yeah, maybe estimate.
Estimate, exactly. That's the word. Sorry. So he combines the coordinate with binary data, and then he estimates the pathogen's distribution by like density. He combines the proximity of each coordinate and then how it works. But kernel is just the name. It's not related with kernel like the grain.
Kernel. Sorry, but he did all that work. By the way, just one more thing. Tarcísio is looking for a PhD position, so if you guys are hiring, keep that in mind. He's a great student. You can see he did all that work by himself. So,
Katia, I have a question. Can you hear me?
yes.
Great job, great work on many different crops there. I have a question about the fusarium that you're finding in the soil in sugarcane fields. Do you know which species of fusarium it is?
So we kind of have some idea because Hardev students is working on that as well. She collected samples here at the station, and also she collected samples on her experiments, but just to commercial fields. So it's not as many commercial fields as we did. So for our work, the one we presented here, we have not done the molecular analysis yet. We are just doing it now, so we don't have data. But yeah, but based on Hardev's student data, it's Larissa, do you remember? It's verticilioids and the other one is it's two species, verticilioids and I think it's vigoriform. I can send you by email.
All right, thank you very much.
The one that she has, and I can tell you the locations where she got it. And once we get the result that's coming this coming week, I can also send you that information, no problem.
Thank you.
Yeah, no problem. Any other question? Oh, we have one more question here. No, the Zoom needs that.
Very interesting work looking for native strains of bacteria to control Cercospora. Are any ideas or any interest in looking at fungal strains to possibly combine with the bacterial strains to look at the microbiome to see if maybe more could be better?
Absolutely. That's on the pipeline. I have an entire collection of fungi that I can test. I just started with the bacteria first. I have 100, thinking total just under 200 isolates to test. So once we know which ones are the best candidates, we're going to start maybe playing around, combining them and looking at an artificial microbiome. But that's later on.
Very interesting. And then just to add another question about looking at controlling Fusarium race 4 in the soil, looking at maybe more of a biological, biostimulatory kind of program to aid the microbes in the soil, to be able to beef them up faster. Any interest in that area? And that would be, yeah.
Yes, we just don't have the resource. So if you are interested, we can talk. I think we have one more question here.
Can you hear me? Yeah. So with the red rot, which is the principal way that gets like the plants get contaminated with it? Because I know that usually borders like inside the system of sugarcane crop are the main reason why you get like red rot inside the sugarcane. But is there any?
It's wind by crop debris.
Oh, okay.
Yeah. Right?
Yeah. So the way I understand, I'm not an expert on that disease. We are learning a lot, but the spores are airborne. Wind, no, the water splash and then windborne. They land on the leaf surface and then they go to that midrib, right? And the leaves are like this. So they will sporulate there on the midrib. And then when the rain comes, the midrib is like this angle, let's say 45 degrees for whom is on Zoom. And then the water will wash them to the stalk. So, oh, that's a great disease cycle we made, but I'm joking. But then you can see we try to represent it here, that reddish on the leaves. And then it rains or there is some water, and then they can be washed here and they come to the stalk. They cause that disease on the stalk. And then the disease will come downwards, that infection will come here in the stalk. And I think one of the symptoms in the field is that you can see that green canopy, but then the stalk is all rotten, which I try to represent it here in this letter G. So this is rotten, and then the top actually bends, or you can see it in the field, you know, and then when they come and they harvest, it can actually go to this phase here. So you can see that it stays there. So now when they burn it, it has a very important role because then they burn the pathogen as well. Now in Brazil, they don't burn anymore. So this pathogen is going to survive on that crop debris, right? And that's important. Now with the ratoon and all of that, it can survive. When we were doing the survey in Tarcísio's work, we found Coletotricum on the billets that we were collecting that were just planted. So it can survive that on those billets, and then they plant it, and of course it will grow with that new planting already contaminated. That's my understanding. Okay, we have one more question here from the audience.
Larissa and Katia and all the team, congratulations for the presentation, for the seminar. And I'm so proud of Ishuri with her English. And I'd like to know, for example, about if you discover microorganisms that can help biological control, how is the business plan of the university? You make a partnership with, for example, a company to make a product, or you sell the strain, do you understand me? Because in Brazil, we launch a inoculant compound by two bacteria. We isolated two bacteria to improve phosphorus use efficiency. And we make a very good partnership with a company called Symbiose. And now this product is here in the United States with the name Solubifos. And I'd like to know how this works in the university.
So I'm going to split that answer with Larissa. But when we wrote that proposal, we already put it all there. So the first thing we want to patent that, right? But then I'll let Larissa talk about UF innovation.
So UF has a IP program. So this would fall under intellectual property from University of Florida. So the first thing we would need to do is to patent whatever, if it's a single strain or whatever product we come up at the end. And then our goal is to provide that to growers so they can multiply the product on farm. So this would be a collaboration between us and the growers, so that it's like, yes. So that it's like at a first instance, and then we can, if there's demand or if the product takes off, then we can think about scaling. And the university has definitely more rules about like monetary compensation and how to structure that from a business perspective and create actual company out of it. And those specific details, I don't know at this point, but I know there is a specific guidelines for that.
Okay, just a comment, because in Brazil, we have discussed a lot about on-farm production. And of course, here, United States is a more rich and developed country, but in Brazil have many problems on farm production because many contamination. When you have, for example, a high production control, for example, high companies or high group of farms, it's okay. But in the most, Embrapa has some publication that 100% of our product produced on farm was contaminated or with some hazard, how can I say, strains. Because of this in Brazil, this is a problem for us. And we try to put some information about the importance to have a higher level of production control. Okay.
Absolutely. Thank you for your comment, and then we'll talk more about it. I think people are ready for lunch.